All right. Well, thanks for inviting me to your foundation support group. It's a pleasure to join you and hopefully share some information about the genetics of PKD. I put this presentation together with the view, and I've been asked specifically to dial back a little bit and maybe present some of the basics of genetics and the genetics of PKD, just so that everyone's all on the same page. Everyone's at different levels in terms of potentially their understanding of the condition or how the inheritance of the condition works, for example. So for some people, this presentation may be a little basic and you understand all of this stuff, and I apologize, and just bear with me while I go through it, so we can get everyone all on the same page. And I'm happy, Nathan, anyone else here this evening, to answer questions as we go. So please feel free to just jump in at any point and ask me questions. If there's anything about the accent that makes it not clear what I'm talking about, feel free to jump in there as well, and I'll gladly repeat what I've said. And if for anyone who's listening to the recording, if you do have questions later, there may be an opportunity for us to get together and hopefully answer them. But a little bit about myself. I'm a genetic counsellor, and I have been practicing for about 10 years now. I graduated from the Sarah Lawrence College in New York back in 2012. And I worked for 5 years in academic medical centres, New York Presbyterian Hospital, and then Children's Hospital of Colorado for 5 years collectively. And for the past 5 years, I've been working with informed DNA. And basically, I focus on neurogenetics, but I also basically see any body system. I talk to families about the genetics of any body system, except cancer, hereditary cancer syndromes. So I see patients throughout the lifespan, from infancy to individuals in their 70s or 80s, and we talk about the genetics of various disorders. And I am very happy to be a part of the life sciences team, and we work with various programs to help patients like yourselves understand the genetics of the particular condition, and also make connections with clinical trials that might be going on in specific programs that might be getting launched. So it's exceptionally rewarding to do what I do, and I always see the job that I have as a privilege to get to walk this journey with patients, even for a short period of time. So enough about me, a little bit about Informed DNA, the company itself. We were one of the largest, if not the largest, telemedicine genetics company out there. We have over 14 years experience with telemedicine. I know in the past couple of years, doctors and doctors' offices have taken to Zoom and phone calls to talk with patients. We've been doing it for a long time, and it's been working just fine. And we have a large team of genetic specialists, around about 100. We've got over 100 staff, around about 100 specific genetic specialists. And we cover all specialties, from cancer to cardiac to neuro to PKD, ocular genetics, reproductive genetics. We have specialties, or GCs that specialise in all the different specialties. And we see over 2,500 referrals, or we receive over 2,500 referrals every month. And these last two buttons down here at the bottom, patient satisfaction and provider satisfaction. Maybe Laura's part of this number here. In fact, that she asked me to come and talk with you. We have consistently year on year been getting good feedback from patients. They like what we do, they like how we do it, and we also get good feedback from providers as well that we work with. Now, what is genetic counselling? I get asked that all the time. People, you chit chat with people and they say, "What do you do?" And you say you're a genetic counsellor, and they always give you a kind of a what? What do you do? And they think we are counsellors, like a therapist or a counsellor who's providing counselling around genetics, and it's kind of what we do. But basically, we help patients and families understand and adapt to psychological implications, but also the medical and the familial implications of hereditary conditions. And there's various things that we do. We discuss with families how diseases are inherited, how different conditions can run in families, and in which mode of inheritance genetic conditions can run in families. We spend quite a lot of time gathering both personal medical histories as well as family histories, and we make an assessment based on all of that information together. We're always thinking family when we're thinking genetic. And so we gather all of these pieces of information together to talk about ultimately the impact of disease and the occurrence of disease in a family, as well as the recurrence, the chance of something happening again to someone else in the family, maybe in a future generation. We also review in some detail with patients the genetic tests that are available today. There's well over 70,000 clinical genetic tests on the market. Not every test is the right test, and so we talk about the testing that really is indicated in this case for them in that situation. We spend some time talking about the implications of a genetic test, what it may or may not show someone and what that may or may not mean for them, and make sure it really is something that they want to do and pursue. And ultimately, this helps people make the most informed decisions about their healthcare. And that's ultimately what I see myself doing. It doesn't matter to me if someone pursues genetic testing or not. The goal for me is to help someone and guide someone to make the best medical decision for themselves. What's the right thing for them to do? It might not be the right thing for me to do or the person beside me, but it's going to be the right thing for the patient to do. And so we help them explore that and make that decision. Now, really what we do kind of on the ground day to day is we talk with patients and families both in a pre-test scenario and then in a post-test scenario. So in the pre-test, we're doing that review, we're gathering the information, talking about diagnosis that someone may have, gathering that family and medical history. Spend some time talking about the genetics, the inheritance and the implications of a condition, and then we facilitate the testing. The inheritance and sometimes the implications of the test results, we spend time on that oftentimes up front before the testing's even done, because when we get to the back end and we have the test results there and I'm about to discuss the test results, as soon as I deliver a test result to someone and I say, for example, your test is positive, you have the condition, we don't hear another thing, and so it's useful to explore those things up front so that someone has a handle on inheritance and how this is going to how this may affect them or how it may affect other family members. So to explore those things ahead of time can be useful once we have the test results back. But when we get those test results back, we review them. Oftentimes it can confirm the inheritance that's suspected in a person or a family. And we can provide medical management recommendations if they're available, and we can write up summary reports, share them with the patients, can share them with providers so that everyone's now on the same page and knowing how to best take care of that person with this now confirmed genetic condition. We can also help families talk to family members, share the information. I get asked regularly, well, how do I talk to my family members about this? How do I start this conversation? We can facilitate those conversations, starting off by providing a written letter saying your family member was found to have this genetic condition. We can just start the conversation and start people talking. We can provide support resources. The PKD Foundation is a support resource, and I can provide that resource to other families who come along. Maybe they don't know about the foundation, maybe it's a brand new diagnosis. And so support organisations that are out there, resources to help find providers that specialise in a certain condition. We help families by providing that information to them. And we also discuss the possibility of being in clinical trials or where to find a clinical trial. Not everyone knows where to find that information, so we help guide that process as well. Now, I always laugh when I see this slide. I was asked to give this talk and as I said, level the playing field in terms of the genetics, basic genetics and the genetics of PKD. And one thing that we do as genetic counsellors, when someone's coming to see us, we'll often Google the condition, the concern or the indication, or just things about genetics. And I actually Googled, what is a gene? And this is one of the pictures that come up. And there's many, many pictures that come up like this. And I don't find them overly helpful because you have to go back about 15 steps to try and understand what's this picture actually showing me. I just asked, what is a gene? You know, it's like if you ask Google where's Starbucks, they'll pull up a map and they'll show you where the closest Starbucks is. That's easy. But you say, what is a gene? And this is what we get. So I don't want you to spend too much time on this diagram because I really don't find it very useful. But the bottom line is that this yellow fried egg looking blob thing here is meant to represent one of the trillions of cells that we have in our bodies. And these blue lines in here represent structures that we call chromosomes. You'll find as you start looking up anything to do with genetics, it's a completely different language. And if you don't speak the language, you don't speak the language. I don't speak Portuguese, so take me a minute to try and understand something that was written in Portuguese, and I'd have to look everything up and try and translate it. It's the same with genetics. It's just a different language. Anyway, these blue squiggles in here, chromosomes. That word means nothing really to anyone if you move outside the world of genetics. A chromosome is really just the packaging material within our cells, the packaging material for our genes. And our genes, you can see down here, this squiggly line here that you can see down here, nothing to anyone. The way I think about our genes is that they are a set of instructions. As humans, we all have about 20,000 different genes in almost every cell in our bodies. And the genes really are a set of instructions that tell us everything about ourselves. We've got genes that tell us our eye colour, our hair colour, how tall we grow. There's thousands of genes that tell the brain how to grow and develop and do its job. The heart, the lungs, the liver, the kidneys, all the different body parts have a set of instructions that tell us how to just basically be humans. Now, of all the genes that we have 2 copies of them. We've inherited one from our moms and one from our dads. So we're each uniquely made up, half from mom and half from dad. And all of our genes are made up of strings of thousands of letters. They really are. We look at the sequence of a gene and it really is made up of strings of thousands of letters. And the letters really are just building blocks or DNA building blocks. And I like to think of it as a genetic alphabet in which there are only 4 letters. A, like apple. Some people think I say that like an E, like elephant, but it's A like apple, C, T, and G. And so each gene is made up of strings of thousands of those letters, just in different sequences. A, G, C, C, T, G, A, C, C, T, however the gene is spelled. All of our genes are really like a blueprint. I said they're a set of instructions, and that's what they are. And they provide the instructions to actually make a protein. And it's the proteins in the cells in the body that actually do the work. The proteins themselves, I think of them as being like strands of beads. And each bead on the strand is an amino acid. And so the genes provide the instruction to make the proteins. And the proteins all are like strands of beads that twist together and have different jobs to do in the cells in our bodies. Now, here's the thing that we don't always hear about, but you've maybe heard of the Human Genome Project and sequencing the human genome, and it's a big deal and it's fantastic and it's changing how we do genetics really. But of the 20,000 genes that we have in our bodies, we really only know what about 6,000 or 6,500 of those genes do with regards to their normal job, and therefore what it means when they don't work. So that means that there are 13,500, 14,000 genes in the body. We don't know what they do. We don't know what their regular job, their regular function is, and therefore what it means when they don't work. And so while we have the technology to read through all of our genes and sequence the human genome, we still have a long way to go to understand, well, what does that mean? And what does it mean for human disease? And what can we do to make that better? So we've still got a long way to go. And the bottom line here is that I will never be allowed to retire because we're finding new genes every day. Now, with each of the conditions that we know about, there are different modes of inheritance or ways that genetic conditions can be inherited. I'm not going to spend any time at all really on X-linked inherited disorders or autosomal dominant disorders. I'm going to spend some time talking about recessive disorders because that's what PKD is. It's a recessively inherited condition, or it's inherited in a recessive manner. And again, when we are you look up autosomal recessive inheritance. This is another one of these pictures that comes up. And I often find myself looking at this picture saying, what is this really telling me? There's grey and blue men and women and grey men and blue men. And I don't really know what this means. And I'm a very visual learner. And so I often wonder if this picture means really not a lot to me, and I understand recessive inheritance. What is someone who's hearing recessive inheritance for the first time? What are they seeing and what are they thinking when they're looking at this picture? So I just want to take a couple of minutes, and please forgive me if you fully understand recessive inheritance. Please forgive me, just bear with me. The next slide will be over in a couple of minutes and then we can move on. But I just want to break this down to really show you what recessive inheritance really means for genetic conditions. So if we look at this slide here, let me orient you. A recessively inherited condition means that you have to have both copies of the gene in question not working to have the condition. You hear people say, oh, I have the gene, or I'm a carrier for the gene, depending on what condition they're talking about. With recessively inherited conditions, you have to have both copies of the gene not working to have symptoms of the condition. So what that means is that parents of someone with a recessive condition are both typically carriers of the condition, and they're healthy, and they don't know they're carriers of the condition because they're healthy. They don't have symptoms of the disorder. This top line here is representing mom. The green lines are a gene, it can be any gene that's associated with a recessive condition, and the X here is showing you that this copy of this gene is not working in mom. She has a regular working copy, and that's enough to keep her healthy, and she doesn't have the condition. This piece down here, this is dad. He's got a regular working copy of the gene and a copy of the gene that doesn't work. Any pregnancy between these two people, between two carriers, there are 4 possibilities that can happen. And this is called a Punnett square. You maybe remember this back from high school biology, but if you don't and you've never seen it before, that's what this is. It's a Punnett square. And it basically shows us that, click through, any given pregnancy, 1 out of 4 possibilities, dad passes on a regular working copy of the gene and mom passes on a regular working copy of the gene. A pregnancy is neither a carrier, nor does that pregnancy have the condition. 1 in 4 possibility, the second option here, 1 in 4 possibility, or 25%. Dad passes on non-working copy of the gene, and mom passes on a non-working copy of the gene. This pregnancy will have the condition, 1 in 4 or 25% chance. Now these other 2 options here, 2 out of 4 or 50% chance, dad can pass on a working copy of the gene, mom passes on a non-working copy of the gene, or here, dad passes on the non-working copy and mom passes on her working copy. These 2 options here, the pregnancy is a carrier, either like mom or like dad, but does not have the condition. So any pregnancy, the chance of a couple who are both carriers of the same condition, the chance that they have the condition, the pregnancy has the condition is 1 in 4 or 25%. So 1 in 4 or 25% chance the pregnancy does not have the condition and they are not a carrier. And then there's 2 out of 4 or 50% chance that the pregnancy is a carrier, either like mom or like dad. I was going to ask at this point if there were any questions. So for the few people that are here, are there any questions about recessive inheritance, or have I bored you completely to sleep because you already knew this? Oh, no questions. I've bored you. You're all asleep, aren't you?
Yeah, I was sorry. I was that. That is. I know that for a lot of people, it's like very easy to understand, but to me, it's like so fascinating because I have 3 boys.
Mm-hmm.
one, it doesn't even, he's not even a carrier.
Mhm,
One is a carrier,
mhm.
and one has the gene. So now that you were saying, like, wow, because at the beginning, like, how can that be possible that one doesn't even
Mhm.
have
It's not
anything
a carrier. Yep.
in, but the other one, it's a carrier.
Mhm.
So it's a very fascinating, and I totally forget the question I was going to do about to make about it, the recessive, but so when you're a carrier, this is
Mhm.
what do you have any symptoms at all, or
No,
you just have to be
typically not, Alejandra. Typically, carriers of PKD typically do not have symptoms. That one regular working copy of the gene is enough to keep them healthy.
okay? Okay, because a lot of people was wondering if you've been a carrier, will you be having symptoms or not? So
Typically not, and it's there are some conditions where there's always exceptions. With genetics, as soon as we say never, it always happens, and so we're always reluctant to say absolutely never. Is it impossible? I can't say that, but typically carriers of PKD don't have symptoms because having that one working copy of the gene is sufficient to keep them healthy. Typically. Because I know someone will tell me that they have symptoms, but typically not.
okay.
And
Was
Claire,
was the breakdown of that diagram useful, Alejandra, to see why there is a possibility of having a child that is neither a carrier nor do they have the condition?
Yeah.
Okay. Was there another question?
yeah, Claire, I just want to, I guess, to make it completely clear, every time there is a pregnancy
Mhm.
between this mom and dad, that each of them have a non-working copy,
Mhm.
they have a 25% chance of passing that on. So it does mean, or I'm asking, does that mean that if they have 4 kids, that they could possibly have 4 kids that do have PKD or any other, inherited disease from the bad copies? And there's also a chance that they could all be carriers, and then there's a chance that they could all not be carriers and not have the disease.
Exactly. And it's so mom has, and I don't know if you can see me here in your small window if I'm on the recording at all, or if you're just recording the presentation, but I'm holding my fingers up here. And if we think about it, any given pregnancy, so if we've got a regular working copy of the gene and a non-working copy of the gene, for mom, any pregnancy, it's 50-50 which one she passes on. Like flipping a coin, heads or tails, 50-50 which one she passes on. And the same thing for dad, it's 50-50, either working or non-working, like flipping a coin, heads or tails. And so we have to have, for someone to have the condition, there's that mom has to pass on non-working and dad has to pass on non-working. So there's 2 things that have to happen here. Each pregnancy, and I think this is what you said, Laura, each pregnancy is a completely independent event. So pregnancy number 1, there's a 1 in 4 chance, 25% chance that pregnancy will have the condition. 2 out of 4, 50% chance that pregnancy will be a carrier, and then a 1 in 4 or 25% chance the pregnancy will neither be a carrier nor have the condition. It's exactly what you said. The second pregnancy is a completely independent event, as is the third, as is the fourth. Having 4 children, as you say, you very easily could have 4 children not having the condition or being carriers, 4 children who have the condition, or 4 children who are carriers. Having 4 children is not a statistically big enough number to show the true statistical value. If a couple had, unlikely, but if a couple had 10,000 children, 2,500 of them would have the condition, 2,500 of them would neither have the condition nor be carriers, and 5,000 of them would be carriers. 4 is not a big enough number to show it, but 10,000 would be. And if you think about that, flipping a coin 4 times, you could very easily get 4 heads or 4 tails or any combination. But if you flip a coin 10,000 times, you'll get 5,000 heads and 5,000 tails.
And with
So
my parents, my parents had 4 kids. And I know that my sister isn't a carrier and doesn't have it, and I do have it, and I don't know the status of my brothers, but I just think it's kind of, you know, there's a little humor in the fact that there's 4 of us, and we're showing a graph here with 4.
Right. And you know what at least 2 people are, right? You know who you are on here, and you know who your sister is on here.
Right.
Yeah.
Yeah. Maybe one day we'll know the status of my brothers. I don't know.
And we maybe talked about this, Laura, but knowing your genetic status or that of a sibling tells us nothing about the genetic status of your other siblings.
Right. Exactly.
They're each independent events. And unless you do the testing, you don't know.
Right. Thank you.
Welcome. Other questions?
yeah, Claire, I was wondering if you have in your presentation a Punnett square for somebody like me who has the condition, and then whether they had children with somebody who did not have the condition but was a carrier or was not a carrier. Do you have those other 2? I suppose I guess there's a few other examples, but
there are, yes, and I don't have them in here, Nathan, but that is a great point, and I can add them in, Laura, and send them over to you if it's useful.
yeah, I think I could probably work it out myself from big
And
biology,
you now have the basic page, so you can add in your X's. But basically,
right.
Nathan, when someone has, and it's a great point, and I really should have thought about it, when someone has PKD and they have 2 non-working copies of the gene, that's all you have to pass on, right? So each pregnancy that you only have a non-working copy of the gene to pass on to that pregnancy. And it's 50-50
Right.
which copy you pass on, but they're both non-working. And then if your partner doesn't have PKD and is not a carrier, they've only got working copies of the gene to pass on. So 100% of your children will be carriers from you, as long as your partner is not a carrier or has the condition.
Oh, fantastic. Okay, that's that makes sense. Yeah.
So 100% of your children will be carriers. And then in the scenario when someone has PKD and then their partner is a carrier, it's a 50, so you will pass on non-working. That's all you have to pass on. And then your partner is 50-50 if they pass on working or non-working. So 50% of your children, or each pregnancy, has a 50% chance of being a carrier and a 50% chance of having the condition.
Oh, okay. Yeah, right. 50-50 having it, but there's no chance of not having.
Exactly.
Yeah. Okay. Yeah. Fair enough.
Exactly. This one here, if your partner is a carrier and you have the condition, this is not an option.
Yeah.
Does that make sense?
Yep.
Because you only have non-working copies to pass on.
And so you'd have, in that case, like a 25% chance of having, or is it I guess I'd have to visualize it. Anyways, yeah, thanks. That answers it. I just would have to.
If your partner if your partner has got 2 regular working copies of the gene, 100% of each pregnancy is 100% chance of being a carrier from yourself. And then if your partner is a carrier, it's 50-50 each pregnancy, what copy she passes on. If she passes on working, the pregnancy is a carrier. And if she passes on non-working, the pregnancy will have the condition, sorry.
Oh, right. Okay, good.
That's
That
your
answers
options.
that.
That's
Yeah.
your options.
Perfect.
Now, if 2 people with PKD have children together, 100% of their children will have the condition.
Yeah, that makes sense.
Does that make sense?
Yep. Yeah, because they don't have the working gene to pass on.
Exactly,
Just
exactly. Neither of the two parents have the regular working copy.
right. Great, thanks.
You're welcome. Other questions?
Actually, I was going to ask the same thing that Nathan did, but you already answered it. Thank you.
And I will put those slides together. This slide is easily to manipulate, so I can share that with you. That is no problem at all. Okay.
I did want to ask the question because I know what is it? Random mutation can lead to disorders as well. Otherwise, we wouldn't have many disorders we have today. Is there any way? Is there? I guess I don't know how. Do we know how often like a random mutation
A random
that leads to
mutation
pyruvate
happens.
kinase deficient symptoms that isn't one that the parents had before, but still leads to the same disorder, I guess?
I
Do you know how
don't.
often that occurs or no?
It depends. Different conditions can have a higher chance of a random genetic event happen. There are some regions of instability within certain genes. I don't have the exact number for the PKLR gene. Doesn't mean I can't find it. I can have a look and see if that data is available. It's not always available, but in the scenario where we know someone has PKD or there is a family history of PKD, we would always recommend doing partner testing. Person maybe doesn't have PKD, but we just want to look, see if they are a carrier of the condition. And so the couple can make and really informed reproductive decisions, but carrier testing can be done. I don't know the exact number for you mean the brand new random genetic event happening?
Yeah, like a mutation that no one else has, or
Right.
a mutation that's not from the parents. Yeah.
Right, not inherited, yes. And I'll be able to I'll have a look and see if I can find the data to see what % of cases are inherited or versus a brand new genetic change.
So I was just going to kind of add on to that. Is there like specific populations that would, I mean, do we find the recessive traits kind of in, like, I don't know, I suppose like sickle cell anemia,
Right.
pardon me, is more common in specific populations?
Yes, and I'll talk a little bit about that. I have information on prevalence of the condition as well as specific variants that seem to be more common in certain populations. So yes, and I have information on that, so we'll get to it. Yeah. Other questions? All right. So moving on. Speaking of pyruvate kinase deficiency, as we are, this slide, I'm almost embarrassed to put this slide on here, but answer some of the questions that you were asking just at the end there. But I'm almost embarrassed to put this slide on here because you know more, the people that I'm speaking to here know more about PKD than I could ever profess to know. And so I'm going to just glean over some of this information, you know, better than I do. But the first thing I tend to do when I see a condition or we're learning about new conditions or what the symptoms of a condition, hemolytic anemia, hereditary hemolytic anemia. Words in medicine drive me crazy sometimes because there's nothing simple about them. What does hemolytic mean? So I always go and look things up to try and break it down into simple terms for myself, especially when I then have to speak with other people about it. Hemolysis is basically just the breaking down and the premature breaking down of red blood cells, which ultimately leads to an anemia. The symptoms, you all know this. The symptoms of PKD can be something as mild as maybe having a pallor, maybe a paleness to their skin or jaundice. And there can be extreme symptoms of the condition where there is excessive breakdown of the red blood cells. All of our blood cells go through our spleen. It's like an organ that cleans all of our blood cells, and those broken down blood cells basically get caught up in the spleen, and that can become enlarged. We know that our red blood cells have iron in them. We need the iron in there to carry oxygen around the body. So if the cells are getting broken down, then we can have excess iron in our bodies, and we don't have a natural mechanism to dispose of excess iron. So that can start to build up in these organs. The symptoms can be mild to severe, as you know, and can require regular blood transfusions. And to answer that last question that was getting asked, the disorder is a pan-ethnic disorder. It does affect all populations. We have some numbers about prevalence, and you can see here it can affect anywhere from 1 in 20,000 to 1 in 1,000, depending on the publication that you're reading, the study that was done, the group of people that were looked at. This is a thought, somewhere between 1 in 20,000 to 1 in 1,000 individuals of European descent. We don't have good data on individuals of other backgrounds or other descent. So this is all European descent. We know that this condition, and you probably know this, but the condition PKD is caused by, when I say pathogenic variants in the PKLR gene. Back in the day, and maybe some people still do refer to as genetic changes as mutations. And as a genetic counsellor, and especially a genetic counsellor who's worked in paediatric clinics, mutation is just a word that's removed from our vocabulary very early on in our training. The thought of someone's child having a mutation or being a mutant is just kind of, we're scolded for saying that word early on, so it's not a word that we use regularly. Pathogenic just means problem causing. There is genetic variation and it's problem causing to the gene. In light of kind of all the information that's been on the news about COVID and a pathogen and a virus, people have said to me, what does this pathogenic mean? Does it mean there's a virus or something going on in there? No. In the case of genetics, pathogenic just means problem causing to the gene. There's variation in there and it's stopping that gene from working as it's supposed to. That's what I mean by that. PKLR gene, again, geneticists naming things, it's great. They use acronyms. They just use acronyms, letters. Sometimes they'll even throw in numbers. And this is what we have to work with as genetic counsellors. Genes are really just acronyms. But PKLR is the name of the gene. The gene itself, the instruction, it makes an enzyme called pyruvate kinase, and the pyruvate kinase is the protein in the cell that actually does the job. An enzyme is just a molecule in a cell that basically pushes forward and makes a reaction happen. And so pyruvate kinase is involved in a reaction, if you like. All the cells in the body need energy to do their job. And the energy is produced through the breaking down of glucose, so effectively sugar. It goes through a process called glycolysis. It's a multi-step pathway or process, and we end up with energy at the end of that. Pyruvate kinase, so the actual protein, the PKLR gene, makes a protein, pyruvate kinase, and pyruvate kinase has a job to do in the last step in the pathway that basically produces a molecule called ATP. And we could go on for days. People spend their whole life studying ATP, but it's a really important molecule for energy and for cells to function and do their job and basically work. The way I think about ATP is really like an energy bar or a Gatorade. The cell needs ATP to keep moving and to keep doing the job that it's supposed to have. So if we have the PKLR gene is not working as well as it should, we have less pyruvate kinase present in the cell. If we have less pyruvate kinase present, the last step in the process from breaking down sugar to make ATP doesn't happen as well as it should. And so we ultimately end up with less ATP. So we end up with less Gatorade or less energy bars. And so the cell, without the right amount of ATP, it can be dehydrated, it becomes abnormally shaped, and it's got a shortened lifespan, and the cell gets broken down. Any questions?
Yeah, sorry, probably you already said, but what it means PAN, the PAN population that you were saying?
What was the question there, Alejandra?
Oh, I'm saying for the previous presentation, I think that you said it's a pan-ethnic disorder.
Pan-ethnic. Yes, pan-ethnic,
Yeah.
that just means it affects all ethnic groups the same way, in the same numbers.
Okay,
Yes, pan just means all, and ethnic just means ethnic groups. So all groups just the same. It affects the groups the same. Meaning that anyone of any given background can have the condition. People in
Okay.
Asia, people in Southeast Asia, people of African descent, people of Hispanic descent, people of Jewish descent, etc. Okay,
okay.
it's pan-ethnic. Yep. Any other questions?
Yeah, I have another question. When you say, and maybe that you cannot answer this one, but what it says insufficient ATP leads to dehydration of the red blood cells.
Mm-hmm.
I noticed that a lot of the people with PKD, they are like very dehydrated, and doctors, they always request that they drink more water. If that makes any change, or it's part of the ATP, the
Right.
insufficient ATP, that the body needs more hydration?
It's not that the body needs more hydration, and having more water in there's not going to keep the cell hydrated. Not having enough ATP makes the cell dehydrated, and so it doesn't matter that the cell's surrounded by water. And it's hard to visualize, but to keep separate what's happening at the cellular level in the body versus what's happening just in general to the body. Now, there may be just a symptom in general of PKD where being anemic can make someone more dehydrated or mean that they have to drink more water. Maybe we need to flush through their organs more often is a possibility, and I don't know the answer to that question honestly, Alejandra, but I think they're two separate things. I don't think it's drinking more water wouldn't make the red blood cells be more hydrated. Okay.
Okay, thank you. I just was like, when
Yeah.
you were, I was listening, I was like, oh, that has anything to do with this?
And I can see where you can make that connection. Absolutely, absolutely. And I don't believe it would make a difference to the actual stop the breakdown of the red blood cells, because the cells don't have enough ATP. They don't have enough energy. And
Okay,
the energy is not coming from the water.
Okay.
Okay.
Go ahead.
Question?
Well, I had a question. I'm sorry. So you were saying that it has an abnormal red blood cell shape. So I imagine a red blood cell is basically like a donut that doesn't have a complete hole punched through it.
Mm-hmm.
What would the shape of an abnormal PK person, like what is that, a specific shape, or is it just abnormal?
It's just abnormal, and it's
Okay.
They call it non-spherocytic. So spheral just means like a sphere. So it's kind of that, as you say, get that round donut shape, because there is like a dimple in the middle. The hole's just not punched all the way through. They lose the roundness to them. They lose that round shape that they have.
Okay. Okay, great.
And maybe you're thinking of like sickle cell anemia. It's called sickle cell anemia, and we're getting way off track here, but it's called sickle cell anemia because the red blood cells actually look like the shape of a sickle. They're like a crescent moon.
Like a crescent, yeah.
Yeah,
Mm-hmm.
exactly. They're like a crescent moon, whereas there is no specific shape here.
Okay.
So in terms of genetic testing for the PKLR gene, so we have genetic testing available, and there's different ways that we can approach testing for PKD. If we know the actual genetic cause in a family, maybe someone else has already had testing and other family members would like to have testing done, we can do, and this is in most cases the preferred thing to do, is what we call single site targeted testing. We know it's the PKLR gene, we know the exact genetic variant that we're looking for, and the lab will just zoom in on that exact location on the gene, and they'll say, does this person have that variant or not? It's a black and white answer. It's a yes or no answer. When we don't know what we're looking for, and someone has a clinical diagnosis of PKD, there's a couple of ways we can approach it. One is through what we call sequence and deletion duplication analysis. Sequence analysis is basically the lab just reading through the entire gene, just looking to see how is this gene spelled. Deletion and duplication, they're looking for pieces missing or pieces extra. We can also do genetic testing for PKD in what we call a multi-gene panel. And this is what's offered through the AGIOS program, the Anemia ID program. They offer a multi-gene panel, where the lab is looking at 51 different genes, and they're all associated with different types of hereditary hemolytic anemias. The PKLR gene is on there, but the lab will then read through and sequence all 51 genes, looking for the answer to see if they can identify the gene that's not working. I've had people ask me if for doing genetic testing, what does this mean? Do you need a spinal tap or a biopsy? Genetic testing has come a long way, and we used to really only blood samples or skin biopsies, but we are now able to do genetic testing on blood sample, saliva sample, as easy as spitting in a tube, and even a cheek swab, so a Q-tip to the inside of your cheek. That's what buccal swab is. The lab can extract all 20,000 of your genes, and then they only look at either the targeted site, they know what they're looking for, they can look at the one gene, or they can test a whole panel of genes from that one sample. And when we run genetic testing, there are several results that can come back. If we know the variant that's in the family and we're doing targeted testing, as I said, it's black and white. You will either have a positive result, meaning you inherited that gene that's not working, or you'll have a negative result. You did not inherit the gene that's not working. Positive and negative. When we start reading through and sequencing an entire gene, or we're looking at multiple genes, we introduce this third possibility here, this variant of uncertain significance, the VUS, and this is the bane of genetic counselors' lives. And if we think about this, the more genes we look at, the more chance we're going to find variants of uncertainty. We all have the same set of 20,000 instructions. 20,000 genes make up who we are. We all have the instructions that tell us our eye color, but all of our eye color is slightly different. So even though we all have the two genes or the multiple genes, in fact, that give us our eye color, those genes are slightly different. And it's just regular genetic variation. Some people have straight hair, some people have curly hair, some people have red hair, some people have blonde hair. It's just genetic variation. It's not problem causing. A variant of uncertain significance comes up when the lab reads through a gene. They find that someone has some kind of genetic variation in there compared to what we expect. A different spelling in the gene compared to a typical spelling. And the lab will ask a few questions. Do we know if this is a true genetic spelling mistake, stopping the gene from working, and have we seen it in other people before that have the condition? The answer, yes, we know this is a true genetic error, then it's a true positive result, and they'll report it back as a positive result. However, if the lab don't have enough data to say this is truly problem causing to the gene, but they also don't have enough data to say that that's truly not problem causing, they will report it back as a variant of uncertain significance. And it's not helpful. It doesn't help with a definitive diagnosis. I've got nothing of any real value to test other family members for, and oftentimes it means that individuals are not eligible for clinical trials because it's not necessarily the true reason for them having the condition. Over time, as labs gather more data, either from individuals who have the condition or from the general healthy population, they are sometimes able to reclassify a variant of uncertain significance. In the event of a reclassification, about 80% of the time it will get reclassified as completely benign, regular old genetic variation, and about 20% of the time they get reclassified as problem causing. The lab generally reissues a new report, and then we follow up with families and let them know that we have it. Now, I was talking about the lab reading through the sequence. This is just an example. This is not any gene in question. In fact, full disclosure, I made this up. This is just literally what the lab is doing when it's doing sequencing. It's reading through the gene, looking to see how is the gene spelled. And I have here a number 1, this is position number 1 at the gene, and this is position 39. And so when you get your test result back, and some of you here may have already had genetic testing, when you see on your test report a C dot and then a number, maybe 350, and then a C arrow G, for example, I'm just making this up. That's telling us that at that exact position, 350, you have a G instead of a C. It's because the lab read through the gene and it knows exactly where in the gene you have the spelling difference. They know the letter that's expected to be there, and it's reporting back the spelling difference that you have. And the reason I'm talking about this is that I was asked to discuss specifically different types of genetic variation that can be problem causing. There are many different types of variants within the gene that can be problematic to the gene. And I could work through it here with this example of this gene sequence, but I don't think it's going to be very helpful. What may be helpful to you is to think about it in just in a common sentence. If we take this sentence here, the original sentence, the grey cat ran down the hall. Many people with PKD have what we call a missense variant. And a missense variant really is just one single letter change within the gene. And so the grey cat ran down the ball doesn't really make any sense. It's a missense. There's a letter change and the sentence no longer reads as it's supposed to. An insertion, or an example of an insertion, is the grey-green cat ran down the hall. Well, cat's not green, so there's been something inserted into the sentence and it's making the sentence not really make sense anymore. And a deletion is obvious. This is where there's a piece missing. And a deletion can either be one letter or many letters, but deletion is a piece missing. And then the duplication, the cat ran down the hall. Again, there's only one cat running down the hall. This sentence really doesn't make sense anymore. And then ultimately, a nonsense variant within a gene basically just chops the gene up and it stops it being read. It's much shorter than it's supposed to be. And that's the grey means nothing, right? It's not a full sentence. Any questions on the different types of genetic variants that are possible that cause problems to the gene? Okay. So I have another example here, and I was asked specifically about this type of variant, so I wanted to show you how this can happen. Oh, was there a question? Okay. If the original sentence is the grey cat ran down the hall, if we have, for an example, an insertion and we add in an extra letter here, this letter A, just shoved into the middle of the word. Looks like it's the word cat with just two A's in there, but what actually happens in a gene when there's an insertion, really what happens is that all the letters get pushed along that one spot. So we added in an extra A here, and all the letters, the rest of the letters in the sentence all get pushed along just one spot. So the sentence without that A would still read, the grey cat ran down the hall. But now I have an extra A in here and everything's just been shoved along one spot. This is ran down the hall. This is what we call a frame shift. It's pushed all the letters out of frame. And that can be problematic. It means that the gene no longer is in the appropriate sequence and it no longer makes the right protein because it's been pushed out of frame. And I believe that I was asked to ask the frame shift, or address the frame shift specifically. Does that answer the question if you're here? I think it was you, Alejandro, right? Maybe.
Yes, sorry. Yeah, I was, you know, this explanation, it's honestly really amazing because it gives us a little bit of a much better idea what it means, all this. when it's a missense or insertion at all, any of these variants, did that make change in how the symptoms shows in each person or not?
Exactly. That's exactly why, and I was going to talk about that, and I believe in the next slide or so. And I can go forward to it, but it really does make a difference, because if we go back here, if we think about it, having a letter difference here for B for ball, like ball still is a word. And maybe a cat ran down, maybe it was a really large ball and the cat ran down it. So it's a missense variant. But is it, how problematic is it? Do we still have some of the protein present? Do we just have it in a reduced amount? How much less do we have? If it's a frame shift and it knocks it out completely out of sequence, maybe we don't have any of that protein there at all. And so, yes, there is some data emerging, and I'm going to talk about a recent study that came out just a couple of years ago, but yes, the specific genetic change can have an impact on how mild or severe the symptoms are, potentially. We're seeing patterns and we're starting to understand more about it. We're starting to understand. If you think about a nonsense, well, this gene just doesn't work at all. This copy of the gene just doesn't work at all. And so that's not going to make any protein. On the second copy of the gene that the person has, when they have PKD, do they have maybe a very small amount, much reduced amount of the protein getting made? And so they may have some symptoms, but milder symptoms. If someone has both copies of the gene just completely not working at all, they don't have any of the protein, so they may have more severe symptoms. The answer to your question is yes. And this is the publication I was talking about here, and this answers a question from earlier as well about specific genetic changes. So far, PKD, we know there are over 300 reported disease-causing variants, and the vast majority of them are missense, and a missense is one single letter change, one single letter difference in the gene. These are common variants, and they're of people of certain ancestries. This here, as I was talking about earlier, this tells us that a position number 1529 having an A instead of a G is a common variant. This is what's happening at the protein level. It's just other technical information that's out there. But this particular variant is common in individuals of Northern Europe and the United States. Second variant here, position 1456, having a T instead of a C, appears to be common in people of Southern European ancestry. And then we have a common Amish variant here as well that can be seen in Amish communities. And what you were asking me earlier, Alejandra, sorry, beg your pardon, when you were saying, can the specific genetic change mean that someone has milder or more severe symptoms? What we call that in the genetics world is genotype-phenotype correlation. Genotype is, what does your gene look like? What are the spelling differences that you have in your gene, or the variant that you have in your gene? And what is your phenotype? And phenotype is your clinical symptoms, your presentation. What does the condition look like in you based on the genetic information that you have? And is there a correlation? This study that came out in 2020 examined 257 patients. That's a reasonably large study. And they found that in general, people who had non-missense variants, so something other than a missense, some other type of genetic variation, in both copies of the gene, they had a higher rate of splenectomy, so removal of their spleen. They had a higher rate of iron overload, and they had lower hemoglobin levels after they had their spleen removed. So they had more severe symptoms and they had a higher number of lifetime transfusions. And in the study, they talk about many different challenges to the study. And one of the main challenges is that when they're trying to say, is there a correlation between what someone's genes look like and the symptoms they have? One of the issues that we have is that not all missense variants are equal. And if we think about that and we take that sentence that we were working with, the grey cat ran down the hall. This is a missense variant, right? Over here we spell grey, G-R-A-Y. I call it E, G-R-E-Y. So this sentence to me makes perfect sense. This is not problem causing. And so you can have a missense variant that may have a milder impact on the gene and how the gene functions. But it's a challenge to try and pull the data together by examining someone's gene and what missense variants they have. Any questions? I've dumped a lot of information on you. I appreciate that. So, based on all the information we've talked about, we know about the inheritance of PKD, we know about the gene, the PKLR gene, we know that genetic testing is available for the PKLR gene. So is genetic testing right for you? Come and talk to your friendly genetic counsellor. We can figure that out. There are lots of questions to ask yourself about genetic testing. And I personally view your genetic information as the most personal thing about you. It is what makes you exactly who you are. And so some people don't want to know their genetic information. They know they have the condition and they don't need to know the gene or the actual genetic variant. Some people absolutely want to know the genetic variant. Things to ask yourself. Does it change my diagnosis? Could it maybe help with prognosis? Could I maybe be eligible for a clinical trial? Could I help other people in the family make informed decisions about their own healthcare? And some people come to me and they say, well, oh, I don't know if I should do genetic testing. And what happens if I do the testing? I don't have any symptoms, but what if I'm a carrier? Is that now regarded as a carrier for a pre-existing condition, or will my family members be then thought to have a pre-existing condition? And there's federal laws in place, or a federal law in place that's been there since about 2008. And that federal law is known as GINA, the Genetic Information Non-Discrimination Act. And GINA provides protections with regards to your medical insurance and employers. So neither of those two bodies can discriminate against you because of a genetic test report. Medical insurance companies cannot charge you higher for a premium, and they cannot deny you a premium because of a genetic test report. Now, GINA does not provide any protections at all with regards to life or long-term disability insurances or military personnel. And I can go into more detail about that or if people have other questions about it. But anyway, things to consider when you're thinking about doing genetic testing. First step, I would say, as I said, speak to your friendly genetic counsellor. And there's benefits for different people, depending on who you are in the family, depending on whether or not you're symptomatic and undiagnosed, whether you've been clinically diagnosed but you haven't had genetic testing, whether you've been diagnosed and had genetic testing. We'll still talk with you as well. And then we have unaffected family members over here. So we can provide information and benefits to different patients and family members, depending on where you are in the family and where you are in the process. Just help you navigate why this may or may not be useful for you. And if you did want to pursue genetic testing or come and speak with a genetic counsellor, certainly through the Anemia ID program, you can be self-referred, patient-referred, or you can be referred by your provider. Get the referral, one of our lovely care coordinators will reach out to you and get you set up, scheduled and registered. We'd meet with a genetic counsellor, we'd coordinate testing if you wanted to pursue it, and then we would have a follow-up conversation with the test results. We'd generate a summary report and then send yourself and or your doctor the summary report as well as your test results. So we cover a lot, and it can happen over a span of a few weeks, because the testing process can take about 4 weeks to come back. And there's several places that you can get additional information on this. One is the Agios Anemia ID program. That's the link to their website. I'm sure you're all very familiar with it. This is a specific link to us at Informed DNA for the Agios program, and we have a dedicated phone number as well for that program. And with that, I'll take any additional questions that you might have.
So we have a question from Jason coming in on the chat. Jason, do you want to go ahead and ask that question?
Yeah, I was just wondering if they shared the data. If you do genetic testing, I was letting my brother and his family know that they can be tested, but he was worried about what would happen with his data or information.
Yes, no. So genetic test results are protected under HIPAA the same way any other piece of health information is protected. Now, if you came to us, Jason, and we ran testing in yourself and we identified your genetic variants, and you said my family members are interested, I would just ask for your permission to access your test report, so we know what we're looking for, in family history information. We never disclose anything that's ever discussed in any conversation. But outside of, you mean the lab or other providers sharing genetic information? No, they don't. It's like any other piece of health information is protected under HIPAA.
Thank you. I appreciate it.
Yeah, you're welcome.
So, for example, I only had one brother, and my brother passed away a couple years ago. So he, of course, he was never tested for PKD. I know I'm a carrier, and we believe that he may be a carrier or not. We don't know if my parents are carriers. I guess one of them should be a carrier because I'm a carrier, and Jonathan was the lucky one. But is that possible that, for example, my nephew and my niece can get tested?
Absolutely, and that happens often, Alejandra, where we don't, the person who would be key to test, who is your brother, is unavailable for many reasons. They are unavailable. Your niece and nephew absolutely can pursue genetic testing based on the family history.
Okay.
Now, all we can say is when we know your test information, we can test your niece and nephew for that information, and they either have it or they don't. Let's say hypothetically they were both negative. That does not mean your brother was negative. We can't infer anything about your brother's test from your niece or nephew. All we can say is that your niece and nephew are negative.
Okay.
Okay. So there's never no inferring with a recessive condition without testing the individual. But they absolutely can pursue testing so that they know themselves. Are they a carrier? What's the chance that they would have a child with this condition? Absolutely.
And now, for example, we go through anemia ID. I know to me it's a little bit complicated. And can we do it through can we request it as a patient, or we need to go through a doctor in order to get?
Oh, yeah, no. So your niece and nephew could come through Informed DNA, self-referred with the family history of PKD. Absolutely.
Okay. So that way, everybody, not everybody, but like, for example, Jason, he has a son. Can his son be tested too? Or if Jonathan wants to get married later in life, can his wife be tested through an MAD too, or he has to be a different one?
So and is Jonathan your nephew or
My son.
Your son? Yes. So he absolutely, if he doesn't have his genetic testing already, can come through anemia ID. And then if he is found to test positive, either be a carrier or have the condition, then his remind me, Sarah, can his partner also come through anemia ID?
The partner, the unrelated partner,
Mhm.
no, but any
Okay.
biologically related
Biologically. Right.
individuals can undergo counseling and testing at no cost through the Anemia ID program.
Okay.
But your
But
sons, sorry,
partners
Sarah.
could have testing through, through their doctors.
Yeah.
Okay.
I was just gonna say
Yeah.
that.
Yeah, I was right because my son, of course, he has PKD, and when he gets older, if he wants to get married, it will be nice to know if the person who he is going to marry get a genetic test done too.
Right? So they're fully informed, right? And
Yeah.
if she's not a carrier or doesn't have the condition, then they know that going in before they have any children, that their children would be a carrier from your son, but not have the condition.
Yeah.
Yeah. Other questions?
So Claire, I was going to ask, you said that if there was a sibling who was unavailable to have the genetic testing, could their children, you answered yes. Does unavailable include unresponsive to, like
Yeah.
they're like,
Yeah.
they never respond, because I have it, and we know my sister, my sister tested and she's not a carrier, but I have two other siblings, and one of those siblings has children, and one of his children has children. So, but he was unresponsive to me saying, hey, you can get this genetic testing because you're my sibling, and there was just no response. But I wonder, if I reached out to my niece and nephew, would they still be able to get the test, even though their father, my brother, didn't really,
Yes. And it comes down to again, the person who would like to test, politely saying they're unavailable to test because they're just refusing to test, and that happens. They don't want to know their genetic status, and that's fine. But their kids absolutely can pursue the testing. Now, obviously, they would need to be an adult and autonomous in making those decisions. If their parents not interested in testing, I don't imagine they're going to want to test their children. So once the kids are adults and autonomous and making their own decision, then yes, they can pursue the testing because of your test information.
Okay, yeah, that's good to know. And they are both adults there.
Okay.
So that's
Okay.
Thank you.
You're welcome. Other questions?
Nathan, I think Nathan was going to ask something.
No, actually, Laura asked the exact question I was going to ask. My older brother has PK as well, and he has some younger kids, and he's not interested in doing too much. But I wanted to offer it to his children who are under 18, but 16 or 17, in that age where they, would be interested in knowing that. It would require one of the parental units in that structure to, anyways, they would have to get permission until they turned 18, right? That's the thing.
Yeah.
That was my question.
They would. Yeah. But then if they turn 18, Nathan, they could use your test information to pursue the testing themselves.
But for example, I think the Nathan question, if the mother is agree, can they still pursue it?
Yep.
Okay.
And I'm not saying my brother would not. I was just saying if he did not. I'm not saying he's not like that. It was to satisfy my curiosity.
I know. You know what? Like I said, I not like and I was also saying my brother passed away, but my nephew and my niece, they still have their mother. So, and one is an adult and the other one is a minor. I think my nephew is 13 years old. So one is very young, the other one can make her own decisions, but not the younger one. So mom can.
And with genetic conditions in general, so if there's no signs of the condition, and we would be looking to see, is the younger one a carrier? for example, we tend to allow them to wait till they're 18 to be involved in that decision-making process themselves. If being a carrier doesn't change their medical management, they're not showing any signs of the condition at all, there's going to be no changes to treatment, then we allow patients the autonomy to make those decisions about their own genetic information themselves when they're old enough. If symptoms are presenting and there's a concern for the condition, or it's going to change medical management in some way, shape or form, maybe there's screening guidelines for a certain condition in children, then we would recommend doing the testing. If there's no change, then we let them make that decision themselves.
So I think that is kind of a tricky situation, because we know that it's a lot of patients with PKD that they've been diagnosed at a later age.
Right.
You know, we have a patient that she got diagnosed like age 55 or something like that.
In her 50s, yeah.
So I guess like you were saying, to be informed, it's a great way for like our families that are around us. They probably never got any symptoms, or they thought I never had any symptoms until they get older, and they finally realize, oh God, it was PKD, and who was going to know? And a lot of times PKD is being misdiagnosed. They tell a lot of times patients that they have some other disease, like thank God now is anemia ID and Inform ID, that it can have a more comprehensive lab that it can help other patients. But a lot of patients, they are like still in the mist. You know, a lot of people, they don't know what's going on with their lives, or
Right.
if why they are sick.
And that's what I mean with, if there are signs of the condition coming up, we know the conditions in the family, then genetic testing can be done, and it can be done quicker rather than misdiagnosis and over to another doctor and another specialist. We know PKD is running in the family, so we know this gene runs in the family, let's have a look at that. And so we would look at it pretty early on, or we'd recommend that family members share the family history with doctors and ask to be tested for the condition. If there's no changes and there's no symptoms, then typically we let kids make that decision when they're older.
Yeah. And one more question. So after you get the test done and everything, and let's say that you find 2 or 3 mutations, can those mutations be like one missense, not one frame drift, and one as a missense, nonsense, and frame shift, or not, or it has to be all the same type?
No, they can be different. And it's when, so let's say someone on one copy of the gene, they have a deletion, and in one copy of the gene, they have a missense. They are what we would call compound heterozygous. And what I mean by that is hetero just means different, like heterosexual, different sexes. So each copy of the gene is spelled differently. One copy has a deletion, there's a letter or letters missing, and the other copy of the gene has a different spelling at a certain position. So they are compound heterozygous. When someone is just heterozygous, that means that one copy of the gene is a spelling variation in there that's stopping the gene from working, and the other copy is working just fine. When someone is homozygous, homo meaning same, each copy of the gene is spelled the same way, and each copy of the gene has the same genetic spelling difference.
And how common is to have 1 or 2 mutations? What is more common, to have 1 or 2 mutations?
You mean have the same? So in that one study I was talking about, with the large study with 237, the majority of people were compound heterozygous. So they had 2 different spelling differences.
Okay. Yeah, and it still is possible that they can show another
Like a
disease
third.
too, right? Like, for
Yes,
example,
true.
if
Yeah, so
if
there's
they have
two.
PKD and then they can find like spherocytosis, for example, correct? That one person can have another genetic problem, correct?
Yes, yes, absolutely. Having one genetic condition doesn't change your chance of having a second genetic condition. You're at the same general population risk of having any other genetic condition. Absolutely.
Okay.
Claire, I know you and I, you were my genetic counselor, so you and I spoke about this, but I just want to ask it just to, for the people on the call and for the recording. My father has passed, years and years and years ago, so way before that I had the genetic counseling. And, you and I were speaking and I was like, well, if my father is a carrier, then one of his parents must have been a carrier, and my mother is a carrier, one of her parents. And you actually said that my parents could have they could be the ones who were the start of, forgive me for my lack of education on this, but could have been the one with the start of the I'm going to say mutation. I know that's the wrong word, but yeah. So
that's okay. If you say it, I can say it. So you can lead the way on that.
Right. Because I forgot the proper word already. Do I have that correct? Am I remembering that correctly?
Yes, you are. And I think it was Jonathan. Jonathan, right? You touched on this earlier about a brand new genetic variant or new genetic mutation.
Yeah.
And it's where a genetic condition has to start. Well, all genetic conditions have to start somewhere. And so without having your dad available to test, we'll never be able to say for sure, Laura. And so with a recessive condition, typically one copy not working comes from one parent and one copy not working comes from another parent. And there is the chance that one of the parents had two regular working copies of the gene, and one of your copies that's not working is a brand new genetic change only. That is a possibility. We do know that genetic conditions have to start somewhere. There are some conditions where it's almost always passed on from a parent. And there are some, because the genetic variants have just gone back for generation before generation before generation. And there's some genetic conditions out there where we know that through lots of studies, 50% of people with the condition, it's a brand new genetic change in them, and 50% of the people with the condition, it was inherited from a parent. And so it depends on the condition, it depends on the gene. Some genes have areas that are really quite unstable and they're quite prone to new genetic changes. PKLR is not necessarily one of those genes. There are hotspots around the gene where we tend to see commonly where genetic changes can happen, but it's not a gene. The new mutation rate, and Sarah kindly chatted me that information, she was doing her homework, in the background it's about 3%, is that right, Sarah? He's chatting me, it's about 3% of the time it's a brand new genetic change can happen, but it's not very common. Does that answer your question, Laura?
Yes, thank you.
You're welcome. That was right, Sarah, right? You said 3%?
Yes, yep. I pulled that from an article.
Okay.
From a scientific journal.
Thank you. So without having your dad available to test, we would never be able to say 100% for sure. But like just given the chance of it being a brand new genetic change is unlikely, or is low, sorry. One probably came from your dad and one probably came from your mom.
I am my family historian, so I always wonder, it's like, where did this start? And of course we'll never know, right? That's something we'll never know. But I wish I could though,
Go back looking through old hairbrushes trying to find hair follicles on them. Where's grandma's hairbrush?
Right.
Has this been useful? Did this address the questions that you were hoping that it would address?
Yes, definitely. It's a very good presentation, Claire. Thank you so much. And I love the way that you used the sentence about the cat. I mean, that, example I think was just easy for everybody on the call to understand. And I'm sure it'll help, those of us who in our community who listen to the recording. Hopefully that'll help them understand it as well. You know, it was just it was a much easier way to understand, you know, what you were talking about.
Right.
For us people who don't or aren't
They don't speak
very
Portuguese.
knowledgeable, right, who don't speak Portuguese, although Alejandra might, I don't know, but aren't very knowledgeable in genetic speaking, right?
Yeah, exactly. And it's it can be difficult to convey, and it's basically what we do, difficult to convey and interpret this other language over here, this genetics language, in a way that the people who don't move in this world understand. And that's basically what we do. We spend our lives, genetic counselors spend our lives interpreting complex human genetics and helping the people with the condition actually understand what's happening.
Mhm.
And so if I've done a tiny piece of that tonight, then, then yay, I've done my job.
Yes, I do believe you've done your job. And I know I was very happy when I got my results back from my genetic testing that basically, you know, was a definitive answer. Yes, you have PKD, because even though I was diagnosed at about age 10, right around that age, you know, as I went through life seeing other hematologists, they were always like, well, we think it might be this, you know, so it was always some other kind of hereditary anemia. And so it's good to have that test to know 100% that it is PKD and that I don't have any other hemolytic anemias that was suggested to me, you know, by
Over the
by
years.
hematologists. So yeah, I know I, you know, personally am very thankful for the fact that I did get the genetic testing, and thankful for the genetic counseling also by you to help me understand, you know, what I was going to, you know, what the test was going to be and what I was going to, you know, see, and then afterwards explaining the test to me as well. So it's very helpful. I mean, I would encourage, you know, our community to do it.
I have another question that now that you were saying that Jonathan, and maybe Jonathan can help me to remember because he's the genius and he's not me. When he said the genetist or the genetics doctor, he said that he was going to do the whole sequencing.
Mhm.
It's like further more information of the genetics, like more in deep, or what it means exactly.
So did he say the test was it whole exome sequencing, Alejandra? Or was
Yeah.
it just gene sequencing?
Do you remember, John?
I think he was doing the whole genome.
Okay, so the kind of looking at all of your genes.
Yeah.
And when doctors are unsure of what they're looking for, sometimes they will run, we call it whole exome. It's looking at all of the genes that actually have known function, well, they have function, or all the pieces of your DNA that have function.
Yeah.
And the issue with it is that while they're reading through, as I said, they're reading through all 20-odd thousand of your genes, 75% of them, we don't know what they do. And so we can generate the data and see what the sequences of the gene, the spelling of the gene, but it's not going to help us with the diagnosis. And so the first thing that the lab is going to do is look at the genes that are associated with anemia, because that was your symptom. And so they'll read through all the genes that are associated with anemia. They will then sequence the genes that are associated with the heart and the bones and the skin and all of these other different body parts, but that's not what you have going on. The doctor was just casting a very wide net to try and find the answer. And there's a much higher chance of finding variants of uncertain significance, and it can cause more ambiguity than it does answers effectively.
I think that the doctor also was worried because we know the anemia also cause problems with the heart. You can have a lot of heart conditions, and unfortunately in my family we have a strong history of cardiac problems. And the doctor was trying to kind of fall, because my brother is how he died. He had a massive heart attack at the age of 42. My dad already had 5 heart attacks, and I have heart problems. Jonathan has heart problems, so it's like kind of like in there, and a blood disorder as well, that he was just recently diagnosed with kind of a rare blood disorder, and I have von Willebrand, but it's different.
Different gene, different chromosome, whole other different condition. That's another different talk.
Correct. So I have von Willebrand, but he doesn't have von Willebrand. He has what is Jonathan factor 7 and 10?
And clotting deficiency, yeah.
That's a whole other different talk.
Exactly, I know.
Whole other different conversation, whole other different set of genes. So the doctor was looking for several things there, Alejandra, with that large test. Yeah, there's a
Okay.
reason to run it. Yeah, he was looking at for several things.
Okay.
Are we good?
Yes, I think if